Today I will discuss about the uh, enzyme module, which is module 8, BOPTOmetry 203, that is a course code. So before starting this one, what is the metabolic process involved in it, and what are the for the metabolism, what are the enzyme need is there for our body? We have to understand all these things. So just I will thoroughly I will tell the enzymatic, what is enzyme and what is the function of the enzyme. Enzymes are generally nothing but it is a biological catalyst that is speed up the reaction, that is control the rate of the reaction. So it will control the rate of the reaction, whether it will go fast or whether it will be reduced. So generally most of the enzymes are globular in nature. So globular means that is the combination of the tertiary as well as the quaternary structure. Some of the enzymes, especially the ribozymes, which is the RNA species, which is acting as enzymes but which is not as a proteinous in character. Generally enzymes are proteinous in nature. So that is one exception is there, that is a ribozyme which is not at all proteinous in nature. Just I will go the importance. Just I will focus on that importance of the enzyme. What is the enzyme? What is the main important role in our body? Generally, it plays some very important role in our bodies. First of all, the main and the prime important part is a metabolism. So whatever food we are taking, it will go through a metabolic process. For that physiological process, we need enzyme. For the diagnosis purpose also we need, as well as the therapeutic cases also, because the diagnosis and therapeutic cases we have to measure the we are doing the enzymatic assay and we are determining the disease. Now the all biochemical reactions are enzyme catalyzed in living organisms. That is true for everyone. So, by measuring the level of the enzyme in blood, that is a biological indicator, that is a good indicator for any disease, such as myocardial infarction. For that, we have to measure the various type of enzymes, such as for the liver, for some liver disease, we are measuring the HGPT and HGOT. These are the types of enzyme where we can, by seeing the level of that enzyme, you can understand whether the organ is in disease condition or not. It can be therapeutically also, we can use this type of enzyme, such as digestive enzyme. Suppose we are taking more food. So if you are taking some uni enzymes, some tablets are there, some carbozymes are there. So it will go, if you're taking that one from outside, it will go to our digestive system and it will help for the digestion process. It will stimulate the digestion process. So if I'm going for classification, various classifications are there. There are 6 major classes are there. So here we can see that the 6 classes are oxidoreductase, transferase, hydrolase, lyase, isomerase and ligase. So oxidoreductase is nothing but the oxidation reaction, reduction reaction. This enzyme is particularly doing the oxidation and the reduction reaction. Here the enzymatic, here the lactate dehydrogenase, we can see here the lactate dehydrogenase is doing the oxidation. It is participating in the oxidation and the reduction reaction. Whereas transferase, it is doing the transfer of some functional group from one compound to the another compound. So here we can see that that is a D-glucose. D-glucose to glucose, D-glucose 6-phosphate. That is a hexokinase molecule. The hexokinase, what he is doing, he is transferring D-glucose to, it is transferring phosphate group from one molecule to the another molecule. So this is the function of transferase. Hydrolase, so here hydrolase means catalyzes the hydrolysis of a bond. Here sucrase is type of enzyme, it's acting on sucrose and it's participating, it's doing the hydrolysis process. What is the hydrolysis process? Hydrolysis process of sucrose to form glucose and fructose. Simultaneously, lyase, that is also an enzyme. In that lyase, pyruvate decarboxylase, it is an enzyme. it is acting on the pyruvate and it's forming the acetaldehyde and carbon dioxide. So this catalyzes the breakage of the bond without the any type of catalysis. This this will involve some androlases also there that will catalyzes the splitting of the fructose 1,6 bisphosphate to glyceraldehyde 3-phosphate and the dihydroxyacetone phosphate. Now isomerase. Isomerase, by seeing the name, we can understand that is a formation of some isomer. The conversion of one glucose 1-phosphate to glucose 6-phosphate. That is a transfer of the phosphate group from one position to the another position. So in case of glycogenolysis also some isomerase enzymes are involved. In case of ligase, what is doing? It is catalyzing the joining of the two molecules. Suppose DNA ligase, it catalyzes the joining of two fragments of DNA by forming a phosphodiester bond. Now, if I'm focusing on the structure of the enzyme, generally enzymes are mainly based on the two structures are there. One is the proteinous nature, protein part, and the non-protein part. So here you can see that what is a holoenzyme. Holoenzyme is such a complex type of protein, such a complex type of enzyme where protein part and the non-protein parts are involved in. So if I'm focusing on the holoenzyme, from here we can see that the two factors are there. One is apoenzyme and one is cofactors. What is apoenzyme? Apoenzyme is nothing but the protein part of the holoenzyme. That is the apoenzyme, and the cofactors are there involving. So what are the cofactors? Generally, we can see here two cofactors are there. One is prosthetic group, and another one is coenzyme. Usually, the small inorganic molecule or atom which is tightly bound to the apoenzyme, that is a prosthetic group. So prosthetic groups are generally inorganic in nature. Prosthetic groups are generally inorganic in nature. You have to remember this one. And the coenzyme, what do you mean by coenzyme? The large organic molecule which is loosely bound to the apoenzyme, that is a coenzyme. That is a coenzyme or the of the apoenzyme. So if I'm focusing on the structure of the enzyme, generally one substrate will come and bind with the active site of the enzyme. So that binds the substrate as well as the cofactors, and the prosthetic groups contains residue that helps to hold the substrate. So these are the subparts of the enzyme that will also facilitate the substrate enzymatic bonding. Active site generally occupy less than 5% of the total surface area of the enzyme. Generally, active sites having a specific shape due to its tertiary structure of the protein. It is having some specific shape, because if it is not having any shape, that means the substrate will not properly bind with the active site. If the substrate, if the active site position or shape will change, that means if the morphology will change, that means the enzymatic activity will lose. The enzyme, the substrate molecule will not come and not properly fit within the binding with the active site. So the enzymatic activity will completely lose. So if I'm concentrating on the active site we can divide in a two way. One is binding site and one is catalytic site. It chooses the substrate and binds to the active site. And one more is catalytic site. It performs the catalytic action of the enzyme. So here some cofactors also there. Already I have discussed that is a two types of cofactors are there. One is inorganic cofactor and one is organic cofactor. So just we have to remember some name. What is inorganic cofactor and where organic cofactor is acting here. Here hexokinase, here cofactors are magnesium plus plus, or in case of carbonic anhydrase the cofactors are zinc plus plus. So here inorganic cofactors are there. In case of organic, where it is, in case of glycogen phosphorylase requires the small organic molecule, that is pyridoxal phosphate. Pyridoxal phosphate is acting as an organic cofactor in case of glycogen phosphorylase. When glycogen phosphorylase is acting in case of glycogenolysis, that requires this cofactor, that is an organic cofactor. So already I have discussed this one, that is a prosthetic group and the coenzyme. So just you have to remember what is prosthetic group. Prosthetic group is nothing but the inorganic part of the apoenzyme, and the coenzyme is the organic part. Inorganic part which is tightly bound, and the organic part which is loosely bound, right? So this part is completed. That is a basic and preliminary part. Next I will go Next, I will focus on little bit on the enzymatic factors affecting the enzyme. What are the factors? What are the factors that will affecting the enzyme? So here we can see that some various when the enzyme substrate reactions is happening. So first and the major factors is the temperature means the enzyme depends on the temperature, because if you are giving more temperature, the enzymatic structure or the enzyme will lose the structure and it will denature. So here we can see that the effect of the temperature on the enzyme, we can plot, if we could plot a graph, we can understand the increasing the enzymatic activity. If temperature is increasing, the enzymatic activity will increasing up to a level, up to optimum level. So heat energy causes more collision and interaction between the enzyme and the substrate, which reaches optimum temperature, which is close to the human body. That is a human physiological temperature, that is 37 degrees centigrade. After that, after heating means the enzymatic activity will lose. That means it will denature the enzyme with the increasing temperature. So if I'm plotting this graph, so we can find here the enzymatic activity is maximum. After heating, the enzymatic activity is lost. Now, second is the effect of substrate concentration. Substrate concentration, if I'm focusing here, we can see that the maximum substrate concentration, if the enzymatic activity and the substrate concentration is there, so increase in the enzymatic activity at the low substrate concentration, there may be lots of active sites are present that is not at all occupied. This means the reaction rate is low, but reaction rate is low. When more substrate molecule will come and bind with the enzyme active site, then the enzyme substrate complex will be formed. That is the transition state. So this is a transition state, or this is the maximum rate of the enzyme substrate reaction. And eventually, when the increasing the substrate concentration, that means that further that will no effect, because if the substrate concentration is more, enzyme the substrate will not at all come and bind with the enzymatic active site, because all the enzyme active site will be saturated. So no more enzyme substrate complex will be formed. So rate will be optimum. So here we can see that the maximum rate we are finding here. So increasing substrate concentration increases the rate of a reaction. Here we can see, and the optimum concentration of the substrate molecule, all active areas are full and working at the maximum efficiency. So here the graph is going parallel to the x-axis. So any increase in substrate concentration, suppose here substrate concentration if we are increasing means here, so no at all any type of enzymatic rate will be changing, means the enzyme substrate complex will not form, because all enzyme active site will be saturated and there will be no back end site for the substrate. So will have no extra added effect. So no active site will be used for the substrate. Now here two types are there, that is an enzyme activator and one is enzyme inhibitor. That is also prime factor, affecting factor for enzyme substrate reaction. So activator means it will activate. So generally by seeing us only we can understand this will activate the chemical reaction, that is chemical species that can bind with the enzyme and increase its activity. Whereas in case of enzyme inhibitors are chemical species that can bind with an enzyme to decrease its activity. So what is the effect of enzyme on the activator and inhibitor? So in case of activator, it will increase the activity of the enzyme, but in case of inhibitor, it will decrease the activity of the enzyme. For example, here activators, here hexakinase 1 and glucokinase is acting as an activator. It will increase the rate of the reaction. Whereas in case of inhibitor, some drugs, some ribonuclease, some inhibitor molecules will decrease the rate of the reactions. It will decrease the enzyme substrate complex formation. So here also two parts are there, the presence of inhibitors and activators. Some allosteric inhibition also there, some allosteric activations also. So here we can see that the binding site, here it is the active site, enzyme enzymatic active site substrate will come and bind. So it is an inhibitor. Inhibitor means this is a allosteric site. So inhibitor, what is their role? It will inhibit the binding of the substrate. So this is a substrate. Here you can see the active site morphology. So here it is not at all possible for the binding of the substrate. Substrate will not come and bind with this part. So if inhibitor is acting here, inhibitor will bind here. So substrate specificity will lose. So substrate will not come and it will not bind at all with the enzyme active site. So it will alter the active site morphology. In case of allosteric activation, what is doing? This is the altered active site. Now activator will come and bind with this site. So after binding, the active site of the enzymatic morphology of the active site of the enzyme will change. So it will throw substrate will come and bind easily with the active site, because the morphology of the active site of the enzyme will change and it is perfectly fitting with the substrate molecule. So substrate molecule will come and easily bind with the active site of the enzyme. So what is apoenzyme? Here apoenzyme is an inactive enzyme. Activation of the enzyme occurs upon the binding of the organic and the inorganic. So this is a apoenzyme. Here metallic cofactors also there. Here coenzymes are there and metallic cofactors and coenzymes are present here. So apoenzyme is an inactive enzyme. Upon activation, upon binding of the cofactors such as inorganic and organic cofactor, it will bind. When it will bind, then only activation will start. So here also we can see the same thing. What is holoenzyme? We have to understand. We have to follow the definition. The complete complex protein with all necessary small organic molecule, metal ions, and other components is termed as a holoenzyme or the holoprotein. So here already I have discussed that active site and the binding site. So enzyme will come and bind with the active site, and enzyme substrate complex will form. Then the enzyme substrate complex, from the enzyme substrate complex, the product will form and the active site of the enzyme will leave. So this is a pocket. This is a pocket cleft of the active site. So today we have learned so many things. We have learned the enzymatic action, various types of enzyme, various types of factors involved in enzyme. Just we have to remember this type of thing, and after the next session I will continue the rest. Thank you.